Hello, it's a pleasure to present at this session. Today I'm going to be presenting on an exciting new program that's enrolling at Brigham and Women's Hospital and at our sister hospital, Mass General, in Boston. I have one conflict of interest to declare. Some of the data I will present has been partially supported by Biohaven Pharmaceuticals. However, the generation of that data and interpretation of that data was conducted completely independently of the company. So the program I'm going to tell you about really gets to the core of something that I think is at the top of all of our minds, and that is, why can we not change the outcome for our patients with neurodegenerative diseases like Alzheimer's, Parkinson's, or MSA? This week we actually heard some great news about a drug that may change the outcome in Alzheimer's disease, and we hope that this is the beginning of a change in the answer to this question. And yet the problem is vexing and challenging, and certainly there's much to be done here. One of the challenges that we face for these complex diseases is that a one-size-fits-all strategy may not work. That a patient may have the same clinical diagnosis, but that there are many paths to this diagnosis, and that a therapy for one patient may not be the appropriate therapy for another patient, even though they have the same diagnosis. And so our biological understanding of these diseases may be limited, and that could be creating a barrier for therapy. Or the way we conduct clinical trials may be limited. And that is that we're grouping together patients who may have different types of biology, and therefore, even if the drug worked for a small group of those patients, we would never be able to know because they were all mixed up. So this is the challenge that one way or another we're all facing, whether we're doing basic science or clinical trials. We're thinking about these problems. So here's an example. Samaya Nazeen's a computer scientist in my group, and she started taking a look at the kinds of data sets that we would like to have in the next few years for MSA, but that we don't have now. And this data set was generated for Parkinson's disease by the Michael J. Fox Foundation and their so-called PPMI cohort. It's a remarkable study in which they have tracked longitudinally hundreds of patients from the time early after their diagnosis for many years. And they've collected biospecimens, clinical data, and it's really been a tremendous boon to the Parkinson's community. Now, if you look at these patients, they are very different from each other. There are some really remarkably different behaviors. So in this graph, we're just looking at the way in which patients progress based on the number of years since they were diagnosed. And if you take a look at the right-hand side, you can see that some patients progress really fast and some patients progress really slowly. And you can imagine if you were to mix this group of patients up in a clinical trial, you would never really see a difference in these patients in the blue line because they're not going to progress in the course of a clinical trial anyway. And you would hope that you would have more of the patients on the orange line where you would hope to be able to see a difference with a drug. The other issue is that the type of biology driving these patients who are progressing very fast compared to the type of disease biology driving the disease process in the patients who are progressing very slowly may be different. And so this is a really fundamental question that we're trying to address here. How do we take into account this variability among our patients and design appropriate clinical trials? So in a conventional clinical trial, which is a perfectly good way to go about clinical trials and has yielded huge advances for medicine, we are grouping our patients together and giving some proportion of those a treatment and some proportion of those a placebo. And that's a perfectly good way. We randomize our patients to each group, and that's a solid way of figuring out whether a drug is working or not over a particular time period. I think the problem for neurodegenerative diseases like MSA is that if we have patients who are very different from each other, who may even have different causes of the disease, and we group them together, then we might actually lose the signal in a lot of noise and not see any difference between the treatment and the placebo group because we didn't understand the differences between our patients well enough to group them appropriately. In other words, we're comparing apples and oranges. The apples may be responding to the treatment, the oranges may not, and we might miss the signal from those apples, so to speak, within our group. So in an alternative way, and it's also a way that's not without its caveats, is that we take a different approach and we track each patient individually or in a very small, deeply characterized group. And this is much harder to do across centers and at large scale, but it's the kind of treatment approaches we're now seeing in the gene therapy world, including for neurologic diseases. And so I call these an N of few clinical trial, where we take a small number of patients, in this case from our clinic, and we track each patient, that's marked by a single line here, longitudinally over time. And so you can see in this case, there's an observational phase. Let's take this patient here, and there's a progression of the symptoms. The patient's given a treatment and it looks like there may be a response. Then the patient is given a placebo and there's still a progression or not much change. And the patient is given a treatment again and suddenly there looks like there's a response. Whereas if you take this patient over here, over time, this patient, regardless of the treatment, seems to just get worse and worse. So in this approach, each patient is compared to their own history. And this could be a very valuable approach when patients are variable and we don't understand their differences and we want to better understand if the drug we're trying is appropriate for a small subset of patients. So how do we achieve this? And so this is really what we call a clinical trial ready cohort for MSA. Ideally, you want a group of patients who are very carefully followed in the clinic. And so there's a really seamless integration between their clinical care and being observed carefully enough for a clinical trial in the future. And so we have tried to create this approach where clinical trial recruitment is fully integrated with clinical care. And so this observational phase where we track our patients very carefully and try and understand them really happens in the context of their clinical visits. It's just that instead of just addressing the clinical questions, at the same time the patients are monitored and assessed with quantitative data. So if we start to ultimately see that some patients respond to a treatment and don't respond to a treatment, this method only is really valuable if we have approaches that guide us to why these patients are different from each other. We need to understand the biology. So when we think about MSA, we think about it in terms of three major pieces of information that we should be getting from our patients as we track them carefully. We should understand their environmental exposures. We should understand something about the protein that is toxic in this disease, alpha-synuclein, and we should understand something about their genetics. And that may give us clues as to why some patients are responding and not responding. And so this is early days for MSA with this kind of effort, but we need to build the infrastructure so that we can increasingly develop this kind of clinical trial for the disease. Now, this approach is really already bearing fruit in the Parkinson's world, thanks to efforts like I described before from the Fox Foundation. We're starting to see our first clinical trials now being directed at specific subtypes of the disease. And the hope is that those trials will yield, dividends compared to, the trials before where everyone was grouped together. So I want to talk a little bit about the alpha-synuclein, the reason we do focus on alpha-synuclein so much in our group. And that is that even though this patient population with MSA may be heterogeneous, there's one thing we do know for sure. When you do score the diagnosis of MSA, regardless of the cause, in each patient, eventually we know that there is the aggregation or clumping of this particular protein alpha-synuclein. And as many of you will know, this protein aggregates, clumps in different diseases, and the three main ones are described here: dementia with Lewy bodies, Parkinson's disease, and multiple system atrophy. And the idea here is that a normal alpha-synuclein protein ends up folding in the incorrect way and accumulating into toxic forms. And there are many possibilities about which these toxic forms are. And eventually we see in the cell aggregation, and this can be in neurons or glial cells of the brain. We see the aggregation of alpha-synuclein. And in MSA, the characteristic finding is in a type of cell called the oligodendrocyte. In the last few years, there have been incredibly important advances in the study of this protein, and I'm not going to talk so much about them today. Suffice it to say, it seems that this protein folds into different shapes in these different diseases, and that the fold may encode some important aspects of disease progression. And so people have become intensely interested in the differences between these alpha-synuclein strains, as they're called. And so as part of our clinical trial paradigm, we need to incorporate that. And so that's why I drew your attention to the alpha-synuclein centric nature of some of the deeper investigations we do in our patients. And so as these clinical trial unfolds and we start to recruit patients from our clinic with a view to future clinical trial, we gather multiple pieces of information at the same time. In the context of a clinic visit, we elicit the history, but we also measure certain rating scales, for example, the MSA rating scales, so that we can quantitate data. We are increasingly moving towards much more quantitative information gathering about our patients' movements, and we are getting that through biometric studies of our patients. This is in collaboration with Dr. Anupam Gupta at Mass General Hospital, and we think this is going to be a key way of improving the way we track our patients. We use a collection of apps at home, or we have a system called the NeuroBooth, which is set up to evaluate many different parameters of our patients' movement and give us much more detailed information that we can quantitate about their motor skills. So that's the clinical aspect. Brain imaging is very important in our paradigm. But then, to get deeper information, genome sequencing, collecting different types of biospecimens, spinal fluid and skin, and from those specimens we can gather a lot of information, and that includes the amplification of alpha-synuclein. And of course, we are also collecting other biospecimens as well. In our study, we collect blood samples, we collect stool samples, and this is all done as part of major studies based at Harvard called the Harvard Biomarker Study and the CLIMB study, where we can look at various parameters in blood, stool, and spinal fluid. But for our particular studies of MSA, we are focused, for reasons I described before, on alpha-synuclein, and we can actually extract alpha-synuclein that's aggregating from our patients through biospecimens now. And we also collect information through an environmental exposure questionnaire. So recently we put this into practice, and we collaborated with Biohaven. Many of you will know Biohaven conducted a phase 3 clinical trial on a drug called vadipostat that inhibits inflammation in the brain. Now, unfortunately, across a whole population, that trial did not succeed. Many of you know this. But what we did in parallel is we conducted one of our N of few clinical trials for vadipostat to take a deeper dive at an individual patient level to see how vadipostat engaged its target and what the effects might be in our patients. And this work was led by Alain, Andy, and Ariana, who you can see pictured here. And this is how it worked. I'm not going to go through in great detail, but in the course of the last 5 years, with Parkinsonism and ataxia, Mass General Brigham has had over 400 patients. 127 of those scored a diagnosis of possible or probable MSA on clinical grounds. And from those 127, we started to track carefully in the clinic around 70 of those patients. And you can see here on the left that we took a variety of clinical scales, brain imaging, we did some genetic testing, sleep studies, and autonomic studies. We also collected various biospecimens for those patients we were starting to follow more carefully, skin tissue collection to generate alpha-synuclein samples, but also to create stem cells that I'll talk about a little bit later. And we also, especially because this drug was inhibiting inflammation, we used a PET scan to measure inflammation in the brain. So here's an example of a patient who has had this PET scan that is showing inflammation that is really quite severe in the region of the brain known as the pons, and also in the region known as the cerebellum, and some other parts of the brain as well. In contrast, this is a control subject here where the signal is very low. This is a patient with MSA of the cerebellar type, where we see considerable pathology in the pons and cerebellum. At the same time, we conducted automated volumetric MRI on our patients, in which we can segment the different parts of the brain and get a much more quantitative view of how those brain structures are changing over time. Now, we then took 20 of these roughly 70 patients and we put them in an observational phase where these deep dive studies continued over a period of at least 6 months. And then we conducted a treatment phase where we gave the patients the drug vadipostat. Now, some of the patients were advanced. You know, our enrollment was open to really any patient, and because we were really looking at how the drug engages the target as opposed to the effectiveness at stopping the disease. And so some of our patients were very advanced and some of them passed away and incredibly generously donated their brains, which allowed us to create sophisticated stem cell models matched to the brain, which I'll mention in a minute. And this just allows us to very deeply understand how this drug is engaging its target and why it may be different in different patients. If we get more precise knowledge, we might be able to target drugs like this to an appropriate subset of patients. So here's an example of 2 patients that were in this study. Now, in one of these patients, you can see that we had a lot of data over, actually in this case, a period of years, and we were tracking inflammation in the brain. And you can see that the inflammation progressively increased, but when this patient took verdiperstat, the inflammation decreased. Here's another patient where we only had the data during the treatment phase, so just before treatment and just after treatment. And while there's a decrease, it's nowhere near as compelling as when you have the prior history of that patient, the prior information. And that's why these longitudinal tracking studies are so important. And so you can see here in this patient, again, very strong inflammation in that area called the pons. This patient had MSAC, and after the drug, this is a dramatic reduction. Although, interestingly, in this patient, some parts of the brain continue to increase in inflammation, and we would like to understand why that is. It may be very important. And here's another version of this PET scan, again, the one looking at inflammation, showing the pre-verdiperstat data with a lot of active inflammation in the brain. In this case, we're showing the striatum, and then after the drug, there is this reduction. This is a close collaboration with Tarun Singhal in nuclear medicine and neurology, and Jeff Young in neuroradiology. And with Jeff's group, we've been able to track volumetric MRI changes in these patients, and this data is all very preliminary, but there are clearly differences among our patients in how their brain MRI scans over time responded to that drug. We make no claims whatsoever about the clinical effectiveness here. These are just biomarker studies, but we think could lead to insights about the drug and whether the drug could be appropriately matched in the future, or drugs like it, to an appropriate subset of patients. Again, very preliminary data, but enough to convince us that this approach is achievable in the clinic and also worthwhile. And as I've mentioned, one way in which our group can, we think, make a difference to patients is actually create a personalized model from our patients. And we do that through human stem cells. So patients who are tracked in this way give a skin sample from which we make induced pluripotent stem cells. So I've mentioned this in a number of other talks, but we can take a skin biopsy, turn them into skin cells, reprogram those into embryonic stem cell-like cells, and from those we can generate the different types of cells that are involved in this disease. And so in the context of our clinical trial, we have some incredibly generous patients who've given their time, but also their biospecimens. And for those who've passed away, some of them generously also donated their brain, which is invaluable, so that we can compare what we see in the dish, in the iPS cells, with what we're seeing actually in their brain. So here's an example of a patient in which in life we looked at the skin and confirmed that alpha-synuclein that you can see in red in the nerves is accumulating, and this helped us diagnose MSA from this patient. And then this patient passed away, and we examined the brain. We confirmed alpha-synuclein in the appropriate places in the brain, so our diagnosis was correct. And at the same time, we took a skin sample and we reprogrammed them into these cells, which we can perpetuate indefinitely in the lab, that are embryonic stem cell-like cells from which we can make brain cells. And this is in close collaboration, the pathology, with Dr. Mel Feeney at the Brigham. And so we have now ongoing analysis of verdiperstat-treated individuals for whom we have matched stem cells and brain. Our hope in time is that we will be able to do this at scale across large numbers of patients. Take different MSA patients, and whether from their spinal fluid, skin, or from their brain, if they've passed away, we can extract the alpha-synuclein protein and aggregate it in large amounts. We've created a human stem cell model that can rapidly show the kinds of aggregates that we see in the brain. So in the brain you can see images on the right, and we can see these images here in the middle, where we can capture very faithfully, we think, the kinds of alpha-synuclein aggregation that we see in the brain. And so ultimately the idea here is to be able to create these personalized models from our patients that incorporate not only their brain cells but the type of alpha-synuclein that is aggregating in that patient, and hopefully look for drugs that can reverse the pathology. In collaboration with Lorenz Studer, we're starting with Parkinson's disease, but we're developing a very exciting technology in which we can take samples, these stem cells, from up to hundreds of patients, and we can subtype these patients and then actually grow in the dish hundreds of patient stem cell lines in the same dish. And so this really creates a whole cohort of patients in the lab that we can study, where we can actually think of doing clinical trials in the dish. Now, this sounds wonderful and it's theoretically achievable. In practice, it is a difficult process, but we are overcoming the hurdles to really make this a reality for our patients with neurologic diseases. So we're very excited about that. So in summary, we would like to establish a deeply studied prospective cohort of MSA and other patients with related diseases at the Brigham with co-enrollment at Mass General. We want to seamlessly integrate this with clinical practice. Currently, we can do this, we think, for dozens of patients, not hundreds, but we would like to build this up over time. This would allow us to make available preceding clinical information, biomarker and biometric data to better understand whether a drug engages its target. We think that we can achieve a definitive clinical trial design through this method, although it has its caveats. And we want to establish connections with industry partners that are bringing innovative disease-modifying candidates towards clinical trial, so that we can really, as we've done with Biohaven, achieve this with other partners as well. I want to mention again that the program leverages a very significant 20-year-old plus infrastructure at Harvard Medical School for two long-standing studies, the CLIMB study that was originally designed for MS, and the Harvard Biomarker study originally designed for Alzheimer's disease and Parkinson's disease, and a biometric infrastructure that I mentioned with Mass General Hospital as well. Ariana is a contact if you are interested in learning more about what we're doing. I am thrilled to go to work every day with an amazing group of people in the clinic and also in the lab. And we have a diverse group, including in their science, from biochemists and cell biologists, computer scientists, and also, of course, clinicians. Could not do this without the funding, including from the MSA Coalition, and this is really a team effort. It takes a village, and I thank you for your time. I look forward to the questions. Thank you.